Hello everyone, this is Medical Gateway, and in this part we're going to study regarding that how the neutrophils and macrophages fight against the foreign invaders. Okay, so this is how it is. For instance, this is in the pink color, this is some foreign invader. Whenever there is a foreign invader, it will release some chemicals. Okay, when it will release some chemicals, then this releasing chemicals will act as stimulus for the neutrophils which are present inside the blood vessels. So the neutrophils will be attracted to this chemical source, this foreign body. Okay, chemotaxis will take place. Now, so neutrophils are roaming in the blood vessels on this stimulus. First of all, the neutrophils will come to the margins of the blood vessels, which is known as margination. Okay, then after margination, the neutrophils will start rolling over the blood vessel, which is known as rolling, and then they will squeeze out. The neutrophils will squeeze out of the blood vessels, finding a small gap between the two endothelial cells and move out of the blood vessels. Now this typical movement of the WBCs, where they squeeze and move out of the blood vessels, is known as diapedesis. And when they move out of the blood vessels, then by amoeboid movement, they are attracted to the source of the chemicals where the foreign body is located. And when they reach the foreign body, they will do phagocytosis and they will eat the foreign body up. Okay? So let's discuss all these steps. So first step is the chemotaxis. Now what exactly is chemotaxis? So chemotaxis is basically the movement of an organism or a cell in response to a chemical stimulus. So there is a chemical stimulus which can be caused by any toxins or inflamed degenerated tissues or complement complex. Okay, now these toxins are basically released by bacteria, virus, or a parasite. So there is a bacteria, a virus, or a parasite, and it releases toxins. And due to these toxins, there is inflamed, degenerated tissue there. So these toxins and these inflamed, degenerated tissues will act as a stimulus, will act as a chemical stimulus. And the movement of any organ or cell in response to this chemical stimulus is known as chemotaxis. So the first step is chemotaxis. Then we have margination. This is margination. So the process where the WBCs move to the periphery or margins of the blood vessels is known as margination. So after margination, we have rolling over. But when the margination takes place, this WBC, this neutrophil, comes and is attached to the endothelial wall. Now there should be some receptor on the neutrophils, as well as there should be some receptors on the endothelial cells. An interaction of these two receptors, the receptor on the neutrophil and the receptor on the endothelial cells, will do the margination. So in neutrophils, there are integrins, whereas on endothelial cells, the receptors are selectin and ICAM, intracellular adhesion molecules. So integrins interact with the selectin and intracellular adhesion molecules on the endothelial cells, and that's how margination is done. So this is the picture of the Guyton. If you focus here, this is the endothelial cell, and the endothelial cell has this blue-colored selectin, and this endothelial cell has this purple-colored intracellular adhesion molecule 1. So they are interacting with the integrins on the surface of the neutrophil. Okay, and this is margination, and then this is diapedesis here, and then the neutrophil is out of the blood vessels. Okay, then after the margination, we have diapedesis. Diapedesis is basically this one. The diapedesis is basically the squeezing through pores is known as diapedesis. So diapedesis is followed by the amoeboid movement in the tissues towards the chemotactic source. And finally, after the amoeboid movement, the neutrophils reaches close to its to the foreign body, the bacteria, the virus, the parasite, where the phagocytosis is done. Now, phagocytosis is basically the cellular ingestion. Neutrophils basically eat the foreign body. Okay, and then after eating the foreign body, they will break down that foreign body, which will be discussed in the next part. So here we have the steps of chemotaxis, margination, diapedesis, amoeboid movement, and phagocytosis. And we'll further discuss the phagocytosis in next part. See you in next part.